presentation topic is exploring the efficacy of herbal formulations as potent therapeutic agent. These are the contents: introduction, review lecture, objective, methodology, results, discussion, and conclusion. Refer. Abstract. The herbal formulations are gaining global attention as safer, affordable, and alternative for synthetic drugs. The traditional knowledge continues guide discovery and applications for herbal therapies. Challenges: lack of standardization, limited regulation, and need for large scale clinical trials. Introduction. Herbal formulations have been used centuries in traditional medicine system like Ayurveda, TMC, traditional Chinese method, and Unani. Key challenges are standardization, quality control, and dosage, and regulation. Literature review and traditional modern evidence. Traditional uses: Ayurveda, a traditional Chinese method, Unani, folk medicine, herbal remedies for centuries passed through generations. Modern evidence: bioactive compounds, flavonoids, alkaloids, terpenoids, and saponins. Validated effects are anti-inflammatory, anti-cancer, cardioprotective, neuroprotective, and anti-diabetic. Lecture review. Advances: cancer and inflammation, brain health, heart health. Challenges: bioavailability, lack of standardization, and limited clinical trials. Objectives: to investigate the bioactive components present in the selected herbal formulations with the therapeutic relevance, to assess the pharmacological and herbal extract against the targeted disease or conditions, to evaluate the role of traditional medicine's knowledge in guiding modern herbal drug development. Methods and materials: research approach, systematic lecturer review. These are the websites now: PubMed and Scopus. These are the bioactive compounds: HPLC, high performance liquid chromatography.
HPLC is okay. Where did you do all this? HPLC, GCMS, NMR, and FTIR?
Outside.
Madam, she has done the literature survey on these topics.
So basically, she has
On
not done
no,
it.
no, no. It's a
Then why don't you say that it is literature survey? Okay, good.
These are the results. 65 plus bioactive compounds, flavonoids, alkaloids, terpenoids, and saponins. Example: the anti-cancer, anti-inflammatory. Majority of flavonoids, alkaloids, highlighting their therapeutic diversity. These are the percentages.
Yeah, that's done. That's okay. That's understandable. Next week.
Pharmacological potential: cancer, diabetes, cardiovascular health, and neurological disorders. These are the percentages. Safety and toxicity, clinical trials. These are the graphs. Discussion: herbal formulation shows strong therapeutic potential. Bioactive compounds effective in major diseases. Evidences support efficacy but needs more validation. Standardization and regulation challenges. Conclusion: herbal formulations are effective, affordable, and safe. Traditional knowledge plus modern science is equal to a strong validation. Clinical trials and quality control are essential. Promoting roles and complementary therapy in healthcare. And these are the references. Thank you.
It's mainly for purification, extraction of sulfuric. The selected bacillus strains from cultured is production media from G gases was extracted from centrifugation, partially purified through ammonium sulfate precipitate and dialysis for active acids. Optimum pH is 8 and 40 degrees Celsius. Introduction: this research shows on isolation of bacillus strain with high precipitolytic activity. There are classified into two types: endo-PG and exo-PG. Endo-PGs are randomly hydrolysis PGs to release galactonitic acid. Exo-PGs remove single galactonitic acid received from non-reducing end. Endo-PG is produced by fungi, yeast, higher plants, nematodes. Exo-PGs bacterial. Exo-PGs differ from producers activity. Endo-PGs, exo-PGs pH is 4.4 to 6. Biochemical structures: PGs are inverting enzymes with right-handed B-Eclix topology. Active site reduce includes arginine, histidine, and lysine. Catalysis involves proton donation and water attack on glycolytic bonds. Mode of action: PG degradation, pestine chains, aciding in fruit juice, clarification, fiber retting, and plant tissue maturation. They are commonly extracted from extracellular, play important role in fungal pathogenicity, and are extremely distributed in bacteria, fungi, yeast, and higher plants and nematodes, make them crucial for industrial use. Exo-PGs acts from non-reducing end of monogalactanosis, release mono or di-galactonic acid, depends on fungal and bacterial origin. While they do not macerate plant tissues, they are essential for efficient pestine degradation and processing. Applications: they are widely used for improve fruit and vegetable oil, juice, and color and clarity. Enzymes from Aspergillus niger, Bacillus substrates, Aspergillus carbogenus, and other species have applied in clarify juices such as papaya, banana, and apple, and strawberry. Study shows that PG gas activity is crucial in the enzymatic retting flax and other fibers. Methodology: samples are collected from different places. Pristinotic bacteria are isolated from yeast extraction peptone media, screened from enzyme activity and optimum for production. Temperatures and uses. Xylose, casein, these are enzyme productions. Purification: ammonium sulfate precipitate and dialysis are used for partial purification of polyglycolitis. Proteins solubility and precipitate are studied by using homophilic series. Result: one bacillus strain shows maximum PG activity. Optimum conformation temperature and pH depends on enzyme yields. Morphology and biochemical textures are confirmed by
We have written pH is wrong. P should be
pH.
small and F should be in the upper case.
Ma'am, this
Yeah,
is
that's correct.
graphs, temperature and pH. The carbon source and nitrogen source. Agriculture applications plays an important role in fruit ripening, softening and post-harvesting processing. Genetic engineering as used for PG substitutions to extend shelf life. Conclusion. Bacillus divides polygalactonoasis shows potential in food processing and fiber treatment and biology technology. Optimum temperatures provide a cost efficiency product strategy used by waste subtractions. Oil derivatives, microbes, strains produce polygalactonoasis shows high potential for industrial and biotechnology applications. References. Thank you.
See, in your thesis I saw it was
Good afternoon, everyone. Myself Vaishnavi S. My topic is probiotic fermentation in non-dairy beverages, potential for gut health improvement. Abstract. It's a promising research area. There are potential solutions for people with lactose intolerance, those on vegan diet. Vegan diet means plant-based substrate like fruits and vegetables. Those who are lactose intolerant support probiotic fermentation, producing beneficial compounds like short chain fatty acids. It can enhance gut microbiota diversity, improve digestion and support immune system. Growing awareness of gut health and increasing demand for plant-based dairy-free options. These beverages offer a unique opportunity to deliver probiotics lactose intolerant, vegan or looking for healthier alternative traditional dairy products. These are the diagrams. Probiotic beverages research suggests that non-dairy probiotic beverages offer a transformative approach to gut health, particularly for lactose intolerance individuals and vegans. Those who are lactogen intolerance means lactose allergic ಇರ್ತಾರೆ ಮ್ಯಾಮ್. ಲ್ಯಾಕ್ಟೋಸ್ ಅಲ್ಲ ಅಂದರೆ ಮಿಲ್ಕ್ ಪ್ರಾಡಕ್ಟ್ಸ್ ಅಂದರೆ ಆಗಲ್ಲ ಅವ್ರಿಗೆ. ಅವರು ಇದನ್ನು probiotic fermentation ನಾನ್ ಡೈರಿ ಪ್ರಾಡಕ್ಟ್ಸ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಅದರಲ್ಲಿ ಲ್ಯಾಕ್ಟೋಸ್ ಬೇಸ್ ಅದರಲ್ಲಿ ಹಾಕ್ಕೊಂಡು ತೊಗೊಂಡ್ರೆ ಗಟ್ ಹೆಲ್ತ್ಗೆ ಇಂಪ್ರೂವ್ ಆಗುತ್ತೆ ಅನ್ನೋ ಕಾನ್ಸೆಪ್ಟ್ ಮ್ಯಾಮ್. Examples are kombucha and water kefir and other water juices. Kombucha ಅಂದರೆ sugary it's a green tea or black tea with fermented sugar. ಅಲ್ಲಿ ಲ್ಯಾಕ್ಟೋಸ್ ಇಂಟಾಲರೆನ್ಸ್ ಇರುತ್ತೆ, ಅವರು ತಗೋಬೋದು ಮ್ಯಾಮ್. ಎಸ್ ಮ್ಯಾಮ್. ದೆನ್ ವಾಟರ್ ಕೆಫೀರ್ ಸಿಮಿಲರ್ ಟು ಕಂಬೂಚಾ. ಎಸ್ ಮ್ಯಾಮ್. ಯಾರಿಗೆ ಹಾಲು ಲ್ಯಾಕ್ಟೋಜನ್ ಇರೋದು, ಹಾಲಲ್ಲಿ ಲ್ಯಾಕ್ಟೋಸ್ ಇರ್ತಲ್ವ ಮ್ಯಾಮ್, ಅಲರ್ಜಿಟಿಕ್ ಯಾರು ಇರ್ತಾರೋ ಅವ್ರಿಗೆ ಈ ಥರ ಇದರ ಆಲ್ಟರ್ನೇಟಿವ್ ಆಗಿ ಕೊಂಬೂಚಾ ಆಲ್ಟರ್ನೇಟಿವ್ ಆಗಿ ಕೊಂಬೂಚಾ ಮತ್ತು ವಾಟರ್ ಕೆಫಿರ್ ಆ್ಯಂಡ್ ಅದರ್ ಫ್ರೂಟ್ ಬೇಸಸ್ ಫರ್ಮೆಂಟೆಡ್ ಪ್ರಾಡಕ್ಟ್ಸ್ probiotic ಮ್ಯಾಮ್. ಕೊಂಬೂಚಾ ಅದು ಗ್ರೀನ್ ಟೀ ಥರ ಇರುತ್ತೆ ಮ್ಯಾಮ್. Objectives: they explore the use of plant-based substrates, fermentation of non-dairy probiotic beverages. It's a survival stability and functionality or probiotics in non-dairy matrix. To examine the impact of fermented non-dairy beverages on gut microbiota, compositions and digestive health. ಅದರ ಕಂಬೂಚ, ವಾಟರ್ ಕೆಫೀರ್ ಇವೆಲ್ಲ ತಗೊಳ್ಳೋದ್ರಿಂದ ನಮ್ಮದು ಜೀವನ ಆಗೋದಕ್ಕೆ ಅಂದರೆ ಗಟ್ ಹೆಲ್ತ್ ಇಂಪ್ರೂವ್ಮೆಂಟ್ಗೆ ಎಲ್ಲ ಯೂಸ್ ಆಗುತ್ತೆ.
Or
Methods and yes, ma'am. ಎಸ್ ಮ್ಯಾಮ್, ಮಾರ್ಕೆಟಲ್ಲಿ ಇವಾಗಲೂ ಇದೆ ಮ್ಯಾಮ್. ಸ್ಕೂಬಾ ಅನ್ನೋ ಲ್ಯಾಬಲ್ಲಿ ಇವಾಗಲೂ ಕೊಂಬುಚ ಇದೆಲ್ಲ ಪ್ರಾಡಕ್ಟ್ಸ್ ಎಲ್ಲ ಸಿಗುತ್ತೆ. ತುಂಬ ಯೂಸ್ ಮಾಡೋದು ಮಲೇಷಿಯಾ ಮತ್ತು ನ್ಯೂಜಿಲೆಂಡಲ್ಲಿ ತುಂಬ ಯೂಸ್ ಮಾಡ್ತಾರೆ ಮ್ಯಾಮ್. ಕಿಡ್ಸ್ ಕೊಟ್ಟರು ತುಂಬ
data collect.
ಅಂದರೆ ಮ್ಯಾಮ್ ಅಲ್ಲಿ ತುಂಬ ಯೂಸ್ ಮಾಡ್ತಾರೆ ಅಂತ ಹೇಳ್ತಿದ್ದಾರೆ ಮ್ಯಾಮ್ ಅಷ್ಟೇ. ಇಂಡಿಯನ್ಸ್
In that
ಮ್ಯಾಮ್ ಅಂದರೆ ತುಂಬ ಅಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ತಾರೆ ಅಂತ ಅಷ್ಟೇ ಮ್ಯಾಮ್. ಓಕೆ. ಪ್ಲಾಂಟ್ ಬೇಸ್ಡ್ ಸಬ್ಸ್ಟ್ರೇಟ್ಸ್ ಅಂದರೆ ಫ್ರೂಟ್ಸ್ ಆ್ಯಂಡ್ ವೆಜಿಟೇಬಲ್ ಸಬ್ಸ್ಟ್ರೇಟ್ಸಲ್ಲಿ ಇರುತ್ತೆ. ಸಿಟ್ರಸ್ಸಲ್ಲಿ ಮತ್ತು ಆಪಲ್, ಪೈನಾಪಲ್ ಇದರಲ್ಲೆಲ್ಲ ಜ್ಯೂಸ್ ತೆಗೆದು ಫರ್ಮೆಂಟ್ ಮಾಡಿ ಹಾಗೂ ಕೆಫಿರ್ ಆಗಿ ಇದು ಯೂಸ್ ಮಾಡ್ಬೋದು. ರಿಸಲ್ಟ್ಸ್: ಪ್ಲಾಂಟ್ ಬೇಸ್ಡ್ ಸಬ್ಸ್ಟ್ರೇಟ್ಸ್ ಇನ್ ನಾನ್ ಡೈರಿ ಪ್ರೊಬಯೋಟಿಕ್ ಬೆವರೇಜಸ್. ಸೇಮ್ ನಾನಿವಾಗ
contaminated and
ಕೆಫಿರಲ್ಲಿ ಶುಗರ್
probe G
ಬ್ಯಾಕ್ಟೀರಿಯಾ ಮತ್ತು ಈಸ್ಟ್ ಇರುತ್ತೆ. ಅದರಲ್ಲಿ ಬಂದು ಯೂಸ್ ಮಾಡೋ ಮೈಕ್ರೋಬಿಯಲ್ ಸ್ಟ್ರೈನ್ಸ್ ಲ್ಯಾಕ್ಟೋಬೆಸಿಲಸ್ ಸ್ಪೀಷೀಸ್ ಮತ್ತು ಬೈಫಿಡೋಬ್ಯಾಕ್ಟೀರಿಯಂ ಸ್ಪೀಷೀಸ್. Plant based substance. Yes. Yes, ma'am.
Point is they will increase the digestibility.
Yes, ma'am. Future of microbial strains and non-dairy probiotic beverages. ಇದರಲ್ಲೆಲ್ಲ ರಿಪೀಟ್ ಆಗಿದೆ ಮ್ಯಾಮ್. Discussion and conclusion. Non-dairy probiotic beverages offer a transformative approach to gut health. They enhance gut microbiota diversity. They improve digestion and support immune function. Benefits extend beyond gut health. Overall conclusion is if you intake this gut health improve ಮಾಡುತ್ತೆ ಮತ್ತು digestion improve ಮಾಡುತ್ತೆ ಮ್ಯಾಮ್ ಅಷ್ಟೇ. Okay.
Is about microbial fungicide, a promising alternative for disease management. First of all, we will go with the fungicides definition. It is a chemical agent which has the ability to reduce or prevent the damage caused by the plants, and it should the characters of fungicides should be like phytotoxic stability, and it is less toxic to human beings and cattle, and it should be ready for the use, and it should be cheaper and easily transposable. And the types of fungicides, there are two types of fungicides, useful and harmful. Usefuls are like Trichoderma, Bacillus subtilis and Pseudomonas streptomycins, and harmfuls Carbendazim, Captan, etc. Mechanism: it works on the four types: antibiotics, antibiosis, hyperparathyroidism, competition and ISR. Antibiotics, coming to this, these are antimicrobial which kill or directly inhibit the growth of the pathogen, and hyperparathyroidism is attacks the pathogens. Competitions, it beneficial microbial competes pathogen for nutrients, and induced, it stimulates the plants for own defense mechanisms. And this is the overview of a healthy plant if we use the fungicides, because it promotes direct action, primering with biofungicides, and its activation of ISRs will be happening here. And what is the use of fungicides? Like it inhibits the protein synthesis of the pathogen and inhibition of sporulation, destruction of hyphae,
Sites, enzymatic
inhibition of electron transference occurs in the pathogen organism. Literature review, according to like from 2020 to 2025, literatures who have written articles on these are based on microbial, they have said that microbial agents improve plant health and placed on molecular physiological modes of actions and field efficacy formulation advancements. According to Andrade, these are the literature reviews, Andrade, Eisel and all, et al. These are the literature reviews, and objectives is to affect the effectiveness of microbial fungicides, role of beneficial microbes and potential microbial strains and eco-friendly alternatives to the chemical based plant disease management. In this we have taken Aspergillus fumigates as a fungi which helps in plant growth promoters and effective sustainable control agents, diversity and pathogens. And coming to the next second objective, it is helpful in inhibit the pathogens, but also stimulates plant antioxidants, suppression and host immunity enhancement. And coming to third, Pseudomonas fluorescens, which enhance the colonization on stability in rhizophores, pool of biocontrol candidates and have the fungicides formulation. And fourth one is it is eco-friendly alternatives to chemical fungicides, chemical based disease management. Based instead of chemical management, we can use eco-friendly alternatives, and it indirectly contributes to yield the improvement and soil health development, and it will be effective for the plant growth. And these are the examples which we have taken, like Trichoderma, Hazardinium, Bacillus pumilus, Bacillus subtilis. These are the examples of the microbial agents which target the other fungal pathogens and which inhibits the growth of that. These are the result of that. And discussions, overallly it is environmental safety, antifungal metabolite potency, and it has pathogen suppression. And however, challenges persist because of the survivals by using formulation stabilities and enhancement of mechanisms, we can use this microbial fungicides as the alternative for the disease management. Conclusion, overall it is eco-friendly, biologically available, and it is a easy solution for the plant and plant fungal disease management. Moreover, we widely use this Trichoderma hazardinium and Bacillus subtilis because of their cross-functional abilities and the capability of making them ideal candidates for the application of inclusions. And by using the artificial intelligence and predictive modeling, we can use this the scalability of microbial fungicides can be significantly used. So these are the references of the literatures, and thank you. Yes.
My topic is a sustainable alternative to petroleum based plastics. Next are the plastic paradox, and what are biodegradable plastics, the science of degradation, applications across industries, benefits beyond biodegradation, and overcoming the challenges, innovations on the horizon, taking action, a call to responsibility, and a greener tomorrow. The plastic paradox. Plastic pollution poses a significant threat to ecosystems, wildlife, and our this is a very crucial role. So conventional plastic persists for centuries, leading to accumulation in landfills and natural environments. It is a cause for
neighboring countries. We have
the urgent need to transition from petroleum
China
to sustainable alternatives is paramount for the planet. So biodegradable plastics offer a pathway to reduce environmental impact through natural decomposition process. So what are biodegradable plastics? Polylactic acid derived from cornstarch or sugarcane and its application in packaging and customer products. Polyhydroxyalkanoates produced by microorganisms offering versatile properties for various applications, and modified starches blended with synthetic polymers to enhance biodegradability and mechanical strength, meeting industrial and home composting standards, ensuring proper decomposition in controlled environments, understanding and recognizing certifications from organizations like VPI and TV Austria for verification. The signs of degradation: microorganisms consume biodegradable plastics, breaking them down into natural elements like water and carbon dioxide. Temperature, humidity and oxygen levels affect the rate and completeness of biodegradation in different settings. Optimizing conditions in composting facilities to accelerate the decomposition process of biodegradable plastics. Biodegradable plastics can also break down in anaerobic conditions, producing biogas as a renewable energy source, ensuring that biodegradable plastics completely decompose without leaving harmful microplastics or toxic residues. So the applications across the industries, from food containers to shopping bags, nowadays we are using many plastics, so that it is a very effective for our human health. So biodegradable mulch films that decompose in the soil, reducing plastic waste and enriching the soil with the nutrients. So we should use biodegradable plastics. So biodegradable sutures and implants that dissolve in the body, eliminating the need for removal of surgeries. So biodegradable cutlery, plates and other disposable items for events and everyday use, minimizing environmental impacts. Biodegradable filaments for 3D printing, enabling the creation of eco-friendly prototypes and products. So nowadays we are using biodegradable filaments for 3D printing also in our daily life. So benefits beyond biodegradation: it lower the greenhouse gases and emissions compared to traditional plastics, and contributing to climate change and mitigation efforts. Minimizing the amount of plastic waste sent to landfills, extending landfill lifespan and reducing the pollution. Utilizing renewable resources instead of fossil fuels, promoting resource efficiency and sustainability. Composting biodegradable plastics improves soil health and fertility, benefiting agriculture.
To access the
Facilitating a circular economy, where materials are reused and recycled, reducing resources depletion. Overcoming the challenges: lowering production cost to make biodegradable plastics more competitive with traditional plastics in the market. Improving mechanical strength, heat resistance and barrier properties to meet diverse application requirements. Investing in composting and recycling infrastructure to properly handle the process biodegradable plastics. Educating consumers about the benefits and proper disposal methods for biodegradable plastics to promote adoption. Establishing clear standards and regulations to ensure the authenticity and effectiveness of the biodegradable plastics. Innovations on the horizon: developing a new biodegradable polymers with enhanced properties and functionalities for specialized application. Nowadays, ma'am, we are using many plastics for our daily lives, ma'am. Now, some of the persons, they are using tea also in plastics only, ma'am. So it is releasing some microparticles and it is affecting our health also, ma'am. So exploring alternative to bio-based feedstocks like algae, seaweed and agricultural waste for polymer production. Designing enzymes to accelerate the biodegradation process and improve the efficiency of composting systems. Integrating sensors and indicators into biodegradable packaging to monitor freshness and reduce food waste. Implementing closed loops systems where biodegradable plastics are collected, composted and reused in a sustainable cycle. Taking action, a call to a responsibility. Choosing products packaged in biodegradable plastics and supporting companies committed to the sustainability. Composting biodegradable plastics in designated facilities or home composters to ensure proper decomposition. supporting policies that promote the use of biodegradable plastics and incentivize sustainable practices, fostering collaboration among manufacturers, researchers and policymakers to accelerate innovation and adoption, sharing knowledge about biodegradable plastics and their benefits to raise awareness and inspire the action. A greener tomorrow. Adopting biodegradable plastics as a part of comprehensive strategy to address plastic pollution and promote the sustainability, reducing the environmental impact of plastics on ecosystems, wildlife and human health for a healthier planet, creating a circular economy where materials are reused and recycled, reducing resource depletion and waste generation, investing in research and development to drive innovation in biodegradable plastics and sustainable technologies, inspiring individuals, business and governments to take action and embrace a more sustainable future with biodegradable plastics.
For anybody who is affected with the TB.
So, rickettsia is a disease which is caused by
Hiding that majorly affects the patient
bacteria.
to increase or decrease.
Is zoonotic in nature. Zoonotic meaning the bacteria doesn't require humans for its life cycle. It is not dependent on humans, and humans
Take care. They are not taking the medications on timely, so they are more prone. Malnutrition and poor living condition that also affect the
ticks
increasing the
have
volatility.
those infected animals such as mice and rats. They the same ones, if they bite humans, we also get infected. That is why it's called as zoonotic in nature.
Social. So
So there are three major groups.
then the surveillance
Rickettsia,
to undergo
they are
program
pure group, typhus group and scrub typhus group.
education on this topic to eradicate this
Rising.
income laboratory coverage in rural and resources.
The objectives of this study are to review epidemiology, diagnostics, clinical outcomes and management,
Data sharing
and
countries
mainly focus on
global monitoring.
India and
The integration of the digital health technologies for the real-time case
regulatory
study.
capacity.
Government introduced next time.
So methodology includes systematic literature review, as it is not very well known in this country. I have considered articles from 2000 as well.
Patient data,
So-
from how many months it has been,
Studies, National
he or she
Disease Control, Indian Council of Medical Research, and
has been established by the government. Advanced challenges in management is diagnosis.
treatment and surveillance.
See, ultrasound, we can find it.
When we come to epidemiology, globally 1 million
This resistance in the TB strain has to be cultured. For patient sample has to be cultured, and we have to find out the patient
city
in which drug.
and major
To be the bedaquiline, delamanide and pretomanide, all of water and oral regimens.
western states. Even Karnataka is having very high cases of rickettsia, but they are often misdiagnosed.
For the careful monitoring of the patient.
Disease gets highly reported in monsoon and post monsoon, and since it is
Related,
reported, it
high
is
dosage
burden.
side effects of
So as
drugs.
here
Novel
there are three
are promised, but uneven access in high burden countries. Immunology, weak T cell and cytokine
first. As you can see here, all
vulnerability, especially with the
treatment across the country, and
host directed
when a company
strategies and boost the immunity and reduce the reliance on the antibiotic. We need to boost the
they obtain
genomic resistance caused by the mutation like genes of RPOB. These genes will
that is 10
and CAT gene will show resistant to isoniazid.
cases which are totally resisted. Febrile cases means cases with fever.
Sequencing is TAC mutation, transmission chain and
Feature. When rickettsia affects a person, they get fever, headache, algia, which is
public health, socio-economic and surveillance dimensions.
Of rickettsia, which is called as eschar. Eschar is a spot, black
Yes, sir.
colored spot, which can be found on the person's body wherever that tick or mite has bitten the person.
Longer driven spread in the urban areas because of the improper hygiene.
Respiratory distress syndrome and
Face the
is brain fever.
surveillance WHO global found and
So the mortality rate of rickettsia is
that persist in low resistant regimes. And future needs
if 100 people which have gotten rickettsia, if they don't get the treatment, 20 to 30 people of them might die.
cost up to 20% more than the drug
With
TB
using
TB
clean
higher than the 50%.
will be reduced to less than 5%. Significant.
Property long
So when we come to diagnosing rickettsia, how to diagnose rickettsia? There are different kind of methods, and conventional which is an agglutination based test, just like blood grouping, which is done on slide.
these 13 decades of
This wheel Felix test
because they are high costing
and
and high impact in South Asia,
are ELISA, which is enzyme linked
European system,
immunization assay, and IFA, which is
limited settings and latent spread.
when we go to molecular based tests, the polymerase chain reaction LAMP technique, which is a modified kind of PCR technique, which have high accuracy, but their access is limited.
The B
The key issue is rural labs lack tools.
or improper
When I was reviewing these literatures, I found out that there are many places they don't even know that there is a certain disease called as rickettsia also. So they might
relapses.
conventional method as well. So hence in wheel Felix test is mostly used, and next that
Control, geographical burden, high impact in South Asia, Sub-Saharan Africa and parts of the Eastern Europe, transition dynamic, overcrowded resources, leading settings and
Yeah.
18
empirical treatment is critical. Empirical treatment meaning the doctors have to know that if there are certain conditions, they just have to go with their gut feeling and start the
survival for the resistance,
clean treatment.
diagnosis, culture-based methods,
Broad spectrum. Yeah.
plus
Alternative to
digital
doxycycline is azithromycin, which is usually given to children under 8 years and pregnant women.
consistently
If they get treated with doxycycline, their bone and teeth development might be altered. That is the reason
and exert
is preferred for treatment in
control and demand
rate,
urgent action because of the spread rate. Since it is a communicable disease, it will random spread. Then
mortality will
rising prevalence introduce the
less than 5%.
infrastructure delay
So what are the public health challenges in India?
mutation
Which is
drug targeted gene
reporting
activity and the bacterial persistence
people who gets
dormancy leads to the relapses and the continuous transmission. Diagnostic rapid molecular
and
sequencing and the digital adherence to the tool improve detection and the treatment
Many-
but remain unevenly accessible. Treatment challenge
if they are there, then
the cost is high, then we need for the safer drug
when I was saying about conventional tests, the rickettsia is so much underdiagnosed that there is only one Indian based manufacturer which provides these kind of reagents.
and proportionately done.
Not many people know how to control these kind of vectors.
Strategies priority.
Vaccines. No vaccine is available for rickettsia.
So access to the
So conclusion and recommendations would be that rickettsial infections are
and we need to
but they
support the patient for their treatment. Invest the novel drugs.
diagnose
Drugs and vaccine need to be improved to reduce the TB cases. especially the M1. Collaboration needed, government
About rickettsia disease.
has a need, communities
Therapy in endemic areas, as we know, how to go with gut.
that's by the year 2030. Overall message, a multi-level
Research priority
optimized treatment, socio-economic
and vaccine development. If we concentrate on those things, maybe we can overcome this upcoming threat to our Indian population.
one of the there.
Yep. Thank you.
The Central Selection Supply Department is the
In the introduction,
man safety.
to see the what are soil microbes and their
Prevent the healthcare.
microbes are the naturally occurring microorganisms like bacteria, fungi, actinomycetes that improves the soil fertility and enhance the crop productivity. And their functions include they involved in the nutrient cycling, organic matter decomposition and mineral solubilization, and their benefits includes they enhance the nutrient uptake up to 10 to 35%, they improve the crop yield, and they reduce the greenhouse emission. This is the pictorial representation of beneficial role of the microorganisms in the soil. Vermicomposting is one of the important process in the nutrient management. Vermicomposting is the process of breakdown of the organic waste into the simpler ones using the earthworms and some microbial inoculants. Let's move on to the principal mechanisms of phosphorus solubilization by the phosphate solubilizing bacteria. Phosphorus is one of the essential elements found in the soil. It improves the soil fertility, which helps in the plant growth, especially root development, which helps in the photosynthesis and energy transfer. Mainly it is found in the bounded form in the soil. Phosphate solubilizing microorganisms helps to convert the insoluble form of the phosphorus to the soluble forms by producing the organic acids such as citric acid, oxalic acid, gluconic acids. They helps to reduce the pH of the rhizosphere. Rhizosphere means it is the region next to the roots of the soil. And arbuscular mycorrhizae fungi also helps to improve the nutrient quality of the soil.
Team Sunlight
This is next move on to the next slide. This is the interaction with
conclude that the
microbes and the host defense response. Here we will see how the microbial inoculants interact with the pathogenic microorganism for the food, for the resources and the space.
chemical
Moving on to the next slide, we will see the review of the literature. Next we move on to the objectives of the research. Here we will see investigation of microbial role in the nutrient availability. The Rhizobium, Azotobacter and Cyanobacteria.
In my thesis, the topic is actinomycetes as a source of bioactive compounds for the pharmaceutical applications.
Form
One type of bacteria, this is actinomycetes. This is a type of bacteria where I am using that bacteria to grow new some medicines which will help for our upcoming futures. In the abstract introduction, and we will study what this bacteria actually looks like. This actinomycetes
enhancing the hyphal
where this bacteria falls under the gram-positive bacteria, in which this particular the main feature of this is it is having a gram-positive feature. It means that cell wall of the bacteria is thickened. Okay, and this bacteria is found in soil and marine system, which is
in our environment, and it has been recognized as a profile procedure for the bioactive secondary metabolites with the significant pharmaceutical applications. From 2018 onwards, we are using this Actinomycetes bacteria as a source of for the production of the medicines in the different way. Actinomycetes as an filamentous bacteria with the gram positive, recognized for their high G plus C content. It has a special recognition for its G plus C. What do we mean by G plus C? It means the guanine and the cytosines which is present in the DNA. Using this branching structure of bacteria, we also grow agricultural and industrial and pharmaceutical applications. This bacteria grows in the spore, and that spores will grow into the germ and the vegetative hyphae and horizontal hyphae. Those hyphae will further grow into the different species. Those are microbiospore and streptosporangium spores and streptomycins. For utilizing this bacteria from the environment, soil and marine, we do genome mining. In the sense, we will study about the genomic data validation. From this we can gain up to the, we have grown the medicinals such as antibacteria and biocontrol, immunosuspension, antifungal, antivirus and anti-tumor. ಈ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ನಾವು ಏನು ಫಾರ್ಮಾಕ್ಯೂಟಿಕಲ್ ಮೆಡಿಸಿನ್ಸ್ನ ಪ್ರಿಪೇರ್ ಮಾಡಿದ್ವೋ ವಿಚ್ ಆರ್ ದೋಸ್ ಅಂತ ಅಂದರೆ ಬಯೋ ಕಂಟ್ರೋಲಿಂಗ್ ಏಜೆಂಟು ನಾವು ಪ್ರಿಪೇರ್ ಮಾಡಿದ್ವಿ. ಆಮೇಲೆ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ನ ಕಿಲ್ ಮಾಡೋಕ್ಕೂ ಇದು ಈ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಆ್ಯಂಟಿ ಟ್ಯೂಮರ್, ನೌ ಡೇಸ್ ಏನಾಗಿದೆ ಅಂತಂದರೆ ಕ್ಯಾನ್ಸರ್ಸ್ ತುಂಬ ಏನಾಗಿದೆ ಅಂತಂದರೆ ಇಟ್ ಹ್ಯಾಸ್ ಸ್ಪ್ರೆಡ್ಲಿಂಗ್ ವೈಡ್ಲಿ ಫಾರ್ ದ ಟ್ಯೂಮರ್. ಆ್ಯಂಟಿ ಟ್ಯೂಮರು ಏನು ಮಾಡಿದೀವಿ ಅಂತಂದರೆ ಆ ಒಂದು ಆ್ಯಂಟಿ ಟ್ಯೂಮರಿಗೂ ನಾವು ಏನು ಮಾಡಿದ್ದೀವಿ, ಈ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಮೆಡಿಸಿನ್ ಯೂಸ್ ಮಾಡಿದ್ದೀವಿ. ವಿತ್ ದ್ಯಾಟ್ ಆಲ್ಸೋ ಆ್ಯಂಟಿ ಫಂಗಲ್ ಮತ್ತು ಆ್ಯಂಟಿ ವೈರಸ್ಸು. ಆಮೇಲೆ ಇಮ್ಯುನೋಸಪ್ರೆಷನ್ಸ್, ಈ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾಗೆ ಏನು ಕೆಪಾಸಿಟಿ ಇದೆ ಅಂತ ಅಂದರೆ ಪ್ಯಾಥೋಜನ್ನಿಗೆ ಸಪ್ರೆಸ್ ಮಾಡೋದು, ಪ್ಯಾಥೋಜನ್ ಇಮ್ಯುನಿಟಿ ಸಪ್ರೆಸ್ ಮಾಡೋದು ಒಂದು ಕೆಪಾಸಿಟಿ ಈ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾಗೆ ಇದೆ. ಸೊ ದ ನೆಕ್ಸ್ಟ್ ಸ್ಲೈಡ್ಸ್ ಆರ್ ಓವರ್ ಹಿಯರ್ ಈಸ್ ದ ರಿವ್ಯೂ ಆಫ್ ದ ಲಿಟ್ರೇಚರ್ಸ್. 2018ಇಂದ ಏನು ಲಿಟ್ರೇಚರ್ಸ್ ಎಲ್ಲ ರಿವ್ಯೂ ಆಗಿದ್ದಾವೆ. ಸೊ ಇನ್ ದಿಸ್ ರಿಸಲ್ಟ್ಸ್ ಬಂದುಬಿಟ್ಟು ಫಾರ್ಮಕ್ಯೂಟಿಕಲ್ ಪೊಟೆನ್ಷಿಯಲ್ಸ್ ಆಫ್ ದ ಆಕ್ಟಿನೋಮೈಸಿಟಿಸ್ 2/3 ಆಫ್ ಕ್ಲಿನಿಕಲ್ ಆಕ್ಟಿನೋ ಆ್ಯಂಟಿಬಯೋಟಿಕ್ಸ್ನ ನಾವು ಪ್ರಿಪೇರ್ ಮಾಡಿದ್ದೀವಿ. 80+ ಆಫ್ ಬಯೋ ಆ್ಯಕ್ಟಿವ್ ಕಾಂಪೌಂಡ್ಸು. ಆಮೇಲೆ 100% ಆಫ್ ಏನಂತ ಅಂದರೆ ನಾನು ಜಸ್ಟ್ ಹೇಳಿದೆ ಅಲ್ವಾ, ಡಾಟಾ ವ್ಯಾಲಿಡೇಷನ್ಸ್ ವಿತ್ ದ ಜೀನೋಮಿಕ್ ವೇರಿಯೇಷನ್ಸ್. ಸೊ ಫಾರ್ ದಿಸ್ ಮೆಟಬಾಲಿಕ್ ಡೈವರ್ಸಿಟಿ ಆ್ಯಂಡ್ ಅಪ್ಲಿಕೇಶನ್ಸ್ ಬಂದುಬಿಟ್ಟು ಯಾವುದ್ಯಾವ್ದಿದ್ದಾವೆ ಅಂತ ಅಂದರೆ ದ ರಿಸರ್ಚ್ ಐಡೆಂಟಿಫೈಸ್ ಸೆವರಲ್ ಮೇಜರ್ ಕ್ಲಾಸಸ್ ಆಫ್ ಬಯೋಆಕ್ಟಿವ್ ಕಾಂಪೌಂಡ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಬೈ ದ ಆಕ್ಟಿನೋಮೈಸಿಟಿಸ್. ದೋಸ್ ಆರ್ polycadids, non-rhizobium peptides, aminoglycine, athrosylene and microdies. These are the medicines which have been provided, and it is also in the biotechnological integration alli nanoparticle synthesis igu help agutte, genetic engineering igu even the bacteria is helping us for the screening purposes and also the synthetic biology. Conclusion ಬಂದ್ಬಿಟ್ಟು ಈ ಒಂದು ಆಕ್ಟಿನೋಮೈಸಿಟಿಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಆಸ್ ಐ ಸೆಡ್ ಯು ಪ್ರೀವಿಯಸ್ಲಿ ಇಟ್ ಹ್ಯಾಸ್ ಬಿನ್ ಯೂಸಿಂಗ್ ಸ್ಪೆಷಲಿ ಇನ್ ದ ಫಾರ್ಮಾಕ್ಯೂಟಿಕಲ್ ಅಪ್ಲಿಕೇಶನ್ಸ್ ಸಚ್ ಆಸ್ ಅಗ್ರಿಕಲ್ಚರ್ ಇರ್ಬೋದು, ನಮ್ದು ಒಂದು ಹ್ಯೂಮನ್ ಲೈಫ್ ಇರ್ಬೋದು, ಇಲ್ಲ ಅಂತಂದರೆ ಬಯೋ ಕಂಟ್ರೋಲಿಂಗ್ ಎನ್ವಿರಾನ್ಮೆಂಟಲಿ ಬಯೋ ಕಂಟ್ರೋಲ್ಸ್ ಮಾಡೋಕ್ಕೆ ಎನ್ವಿರಾನ್ಮೆಂಟಲ್ಲಿ ನಾವು ಏನು ಮಾಡ್ತಿದ್ದೀವಿ ಅಂತಂದರೆ ಈ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ನ ಯೂಸ್ ಮಾಡ್ಕೊಂಡು ನಾವು ಮೆಡಿಸಿನ್ ಯೂಸ್ ಮಾಡ್ತಿದ್ದೀವಿ ವಿಚ್ ಈಸ್ ವೈಡ್ಲಿ ಯೂಸ್ಡ್ ಆ್ಯಂಡ್ ಇಟ್ ಈಸ್ ರೆಕಗ್ನೈಸ್ಡ್ ಫಾರ್ ದ ಸ್ಪೆಷಲ್ ಫ್ಯೂಚರ್ಸ್ ಆಫ್ ಇಟ್. Thank you.
With significant antimicrobial potential.
Antimicrobial activity about both the pathogens which give you the disease.
Escherichia coli. E. coli. E. coli shows the best. With blue and one. Blue. Sorry, it's just quickly control. This is E. coli. Every time E. coli shows the best.
So what further?
Now we can see single nanoparticles is a vast and broad and rapidly expanding. We can synthesize easily from natural source, plants, soils, like that only we can easily synthesize, and it will be helping medicine, electronics and further use also. We can see in water purification and textiles and other industries.
Photobiological synthesis of zinc oxide nanoparticles from central Asiatic leaves and their antibacterial potential. The study reports green synthesis of zinc oxide nanoparticles using extracts of central Asiatica as a reducing and stabilizing agent. Fresh leaves were taken from
Very good. Very good.
they are taken and washed and freshly it is distilled, then dried, powdered, followed by the extraction with the distilled water. Through their boiling and centrifugation to obtain the plant extract. Then after that heating the plant extract under stirring, resulting the formation of yellowish precipitate. And after that centrifuged, then heated under different temperature. Then the silver nanoparticle sample will be collected. After that synthesis of zinc oxide
So you
nanoparticle.
did both the silver and zinc
Yes, ma'am. But have taken
data on zinc.
So after that synthesis of zinc nanoparticle, then for further examination we have given for DLS, zeta potential, and next broad dilution. Then UV spectrum.
What is the size of the nanoparticle you got?
Ma'am, 0.36 nanometer.
Oh, very nice.
Then FTIR of zinc oxide nanoparticles, then interpretation of FTIR test results with their functional groups, then zone of inhibition in agar-well diffusion.
Which are the pathogen?
E. coli, Escherichia coli, Salmonella typhi, Bacillus ceres, then B. parasites.
Oh, you had all these four pathogen.
Systematically we got this results.
Which is
Then-
the best one?
E. coli.
E. coli. Which is, should explain me the table. What is this thing?
Antibacterial activity measured with zone of inhibition in millimole.
What is this red and
Zone of inhibition is higher level.
what is that? Blue.
Both are same. Blue one is antibacterial activity showing against control. 10 is control. This in Escherichia coli, 10 is the control. Then 4 is the zinc nanoparticles. So here the conclusion we can say that the study demonstrates the green synthesis of zinc oxide nanoparticles using Centella asiatica extract rich in bioactive compounds.
Okay. For antimicrobial, you suggest which is for which pathogen?
Escherichia coli and Salmonella typhi. Both are best. Thank you.
Myself inside for my academic project, I chosen cloning and expression studies of replicase protein gene from monkey yellow mosaic virus in E. coli.
When did you do this?
This is a picture that I practice in Bangalore, GK College, in biotechnology, plant biotechnology. For my guidance, she is assistant professor Punima. I hope you know mugwort. Mugwort is a green grass which is rich in protein. Our Asian has it called summer bee due to its property because it reduces the heat in human body. So they used to call it as summer bee. And this is very rich in protein. So gym students use as a protein. Actually, our mugwort, unfortunately, our mugwort plant is suffering from the viral diseases. That's called the yellow mosaic virus. Which is spread by the very tiny, cute little flies is called white.
You say cute?
Yeah, actually, the wings color is white, but the body of the color of that insect is slight blue. So I call that as cute. as we know, viruses are very different in protein. In that, replicase is a very important protein which helps to virus to multiply the plant protein. So I focus but I choose replicase gene in there. So my work is here. I took replicase gene in virus and I insert that gene into bacteria called E. coli. And I forced that bacteria has protein.
So how did you do these experiments virtually?
Actually, for cloning, I use PCR plasmid and digestion, distributed digestion, and for conformation, I use agarose gel electrophores.
So where did you do all these experiments?
In Bangalore, their lab.
Okay. How many months you took?
Ma'am, only 1 month because of profession. And here for cloning, I use that technique like PCR plasmid and restriction digestion and another. For expression, I use CDS page. In CDS page, I use for protein production to confirm the protein production. By the practicing the average gel electrophores. For expression, I confirm there in my protein STC page to check the protein production. Yeah, it's for that I observe the bands around that 34 kDa. Yeah. For better result and for strong result, I take IPTC 1 mm IPTC for for 37 degrees Celsius for 4 hours. So I got the result. So this proves that we can use that replicase protein as a as an antibody.
There is no work on this earlier. Sorry.
I'm from my father and I'm from farmers. So my grandfather always used to take this virus friendly photo. So ನಾನು ಏನು ಮಾಡಿದೆ ಅಂತಂದರೆ ಲೈಕ್ ಫಾರ್ಮರ್ಸ್ ಇರೋದ್ರಿಂದ ನಾವು ಯಾವಾಗಲೂ ಈ ಕಂಡೀಷನ್ನ ಫೇಸ್ ಮಾಡ್ತೀವಿ ಫಾರ್ಮರ್ಸ್ ಆಲ್ವೇಸ್ ತೋರಿಸಿರ್ತೀವಿ. ಸೊ ನಾನು ಏನು ಮಾಡ್ತಿದ್ದೆ ಇದ್ರ ಮೇಲೆ ನಾನು ಮುಂಚೆನೇ ಡಿಗ್ರಿಯಲ್ಲಿ ಬಂದಿದ್ದೀನಿ. ಸೊ ನನಗೆ ಅವಾಗಲೇ ಥಾಟ್ ಇತ್ತು ಈ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ನಾನು ಮಾಡಬೇಕು ಬಟ್ ಅಲ್ಲಿ ನಾನು ಬೇರೆ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡ್ತೀನಿ. ಸೊ ಇದ್ರ ಬಗ್ಗೆ ಸ್ವಲ್ಪ ಐಡಿಯಾ ಇತ್ತು, ಜನರಲ್ ನಾಲೆಜ್ ಇತ್ತು. ಸೊ ಐ ಚೂಸ್ ದಿಸ್.
Good. All the best.